From Microbe TV, this is TWiN, This Week in Neuroscience, episode number 63, recorded on August 18, 2025. I'm Vincent Racaniello, and you're listening to the podcast all about the nervous system. Joining me today from New York, Tim Chung.
Hello. Hi, Vincent. Hi, Vivian. It's we're getting 63 is quite a big number. I'm getting a little bit scared.
Well, when we get to 100, we'll have to do something in person. We're doing Immune 100 in December, and everyone's going to come here to the incubator.
Oh, okay.
Oh, fun.
We'll probably have a little audience next door, about 20 people, and we'll stream it. That should be fun. Just do something different. Also joining us from New Orleans, Viv Morrison.
Hey, guys. Good to see you again.
Actually, it's Vivian. I'm sorry. I didn't mean to abbreviate.
No. It's okay. Actually, that's what a lot of people call me.
Viv?
Viv's my nickname. So.
Viv is good. It means life.
Yes, it does.
At least
got lots of it.
Got lots of life. Mm-hmm. Well, if you enjoy these science programs where we give you the truth, no disinformation, no lying about science. Oh my gosh, I just recorded a Beyond the Noise with Paul Offit. We went through all the lies of RFK Jr. It could go on for hours. Anyway, we'd love your support to do these. We don't do ads. We just depend on listeners. Microbe.tv/contribute. And you know, if you use the Patreon option, I just added a yearly subscription. All right. So usually people pay every month. And if you do a year, you get 10% off.
Yeah, I saw that in my email.
So a bunch of people have taken it up. That's really nice. It's just a convenience thing, so you don't have to see the charges all over the place every month. I guess it doesn't really matter, but you save 10%. What the heck? And you're supporting science communication, so that's the bottom line. Before we do our paper today, I just wanted to point out, I was looking for some protein data bank files, and I saw over on the protein data bank, you know what that is? That's where you put that's where all the structures of proteins are put. The molecule of the month. So they have this thing called the molecule of the month. It's ARC.
Oh, no way. Shout out to Jason.
It's called activity-related cytoskeleton-associated protein. So it's actually a retroviral capsid that's been repurposed. And this is what Jason Shepard works on. And he thinks it's important for neurotransmission. So it's very interesting. Mm-hmm. It's actually a capsid. It's an icosahedral capsid. You can see in the image there. Of course, it doesn't have any viral nucleic acid, but maybe it has other nucleic acids in it.
That's good.
Is it, when it's internal inside the cell, is it already folded into a capsid structure, do you know? Or is it only when it's outside the cell?
I believe it's a capsid inside the cell, yeah.
Okay.
But that's a good question because... so this has some homology to HIV gag. But HIV, the capsid doesn't mature until after budding. So there's a protease that's packaged with the capsid. So the membrane is around the outside, and then inside is the capsid. But it's not mature. And as soon as budding happens, the protease cuts, and then it matures. So... something different is going on here because these are mature without budding. I don't think they're even in membranous vesicles at all. Anyway, it's pretty cool that they have a nice page on it. You can see compared with other capsid-like molecules as well. So I emailed it to Jason. He said, yeah, I knew that.
Gosh.
I didn't think he prowled around.
Jason, he probably does.
PDB.
Jason probably lobbied for ARC to be.
Yeah, of course. So it's fun I need to make pictures for the textbook, so I go to PDB a lot and get coordinates, and I spend hours making pictures. Oh my God, it's so hard to do. The simplest stuff is easy, but the molecules, it's hard to know what's what. I mean, I spent many days this week making one RNA polymerase.
Can like AlphaFold and all these AI tools, are they not good enough for or accurate enough for this kind of stuff yet?
You know, this is a good question. If you could tell some program, could you please make the structure and color each domain a different color? Yeah, why not? People make pictures all the time with AI. Why can't you make molecules?
Yeah,
right?
exactly.
I don't know. AI is just, I think we all have these like great ideas about like what it could do. And then we have to reflect on like where it's actually at. And it's like, oh yeah, unfortunately, like it's not there yet.
So somebody sent me an email. It was very funny. He said, I found this vaccine scar on my shoulder. I wanted to know what it was. So he said, I asked ChatGPT, and they told me it could be my oral polio vaccine scar. Oral polio vaccine. Come on, it's taken by mouth. Come on, AI.
Wow.
So that's an example. That's
it's so smart and so dumb at the same time.
It's amazing.
Yeah.
Amazing. Well, anyway, I am dumb when it comes to neuroscience. So fortunately, I have Tim and Vivian here because it's my turn to do a paper today. And I picked one that has a little microbiology associated with it. It also has a lot of biochemistry and also neuroscience. So it should please everybody. It even has microglia to please Vivian.
Wonderful.
Right?
I mean, everybody's talking about microglia these days, and the microbiome.
So this is a cell-hosted microbe paper. Lactobacillus acidophilus promotes cognitive function recovery via regulating microglial peroxisomal function in cerebral ischemia.
So we just need to eat lots of yogurt, right? Isn't that what are the live cultures or the cultures?
It's one of them, yeah.
Yeah.
Or you could take probiotics because that's a common probiotic.
Yeah.
But you should. I'm not going to give you medical advice.
Yeah, please don't. That's
after
not what
reading
we're here for.
After reading this paper, you might want to look into Lactobacillus acidophilus because, as far as I know, it's harmless.
If it's in yogurt, then it's actually fascinating. We'll talk about it in a bit.
It is in yogurt. Yeah, I bought
Maybe
some Greek yogurt. Yeah, that's true.
It may not be enough.
And I think it might also depend on what you already have in there.
Yeah.
I would point out, because Vincent's going to get into it, but the paper is from a Chinese group. And if there's one thing Chinese people is famous for not eating,
it's
yogurt. That would be yogurt.
Yeah. They don't like yogurt?
It's just culturally not a thing.
Mm-hmm.
That's a
not
play
yet
on words.
But I think people are...
I love it. Tim, did you get that?
I wasn't listening.
It's a cultural thing.
Yeah,
that's a good one. All right, folks, that's it. Thank you. We'll see you next month.
So, it's from Wuhan and Hubei. And the first two authors are Yimin Huang and Shisheng Zhang. And the last author is Huaxi Zhang. And this is all about cerebral ischemia, which is a kind of stroke, right? Mm-hmm. That happens. Insufficient, as they say, cerebral perfusion. You get a lack of oxygen, folks. Mm-hmm. You get hypoxia, nutrient deprivation, often an occluded vesicle, right? So you're going to get that. You can have it from atherosclerosis. You can have it from hypertension, Moya disease. I don't even know what that was.
I don't know what that is.
Hereditary arteriopathies or vasculitis. So a lot of things can cause cerebral ischemia.
Yeah, but your neurons are super duper sensitive to lack of oxygen because they need so much energy. Yeah.
So that's why in COVID, if you have severe COVID and you're not getting enough oxygen, like below 92 or whatever, O2 sat, you can get cognitive issues. Mm-hmm. It's not because the virus is there. It's because you don't have enough oxygen. Mm-hmm. Plus you have inflammation. As you will see today, inflammation is not a good thing either. So what happens when you get cerebral ischemia? You can get transient issues like neurological disability, vascular dementia. That That's the second most prevalent etiology of age-related cognitive decline. So 15 to 30% of all dementias are
that
these.
was kind of blew my mind. I was like, wow.
Or if all we need is just more blood to the brain.
I had a colleague at Columbia. He's still alive, amazingly. He had a ruptured aneurysm in his brain.
Oh dear.
And he's not recovered a lot of motor functions, but he's got a normal life. And he was just in therapy for years. It was they wrote him. The docs wrote him off.
Yeah, I mean, so that's the hemorrhagic, or it's an aneurysm, right? But you mentioned there's ischemic stroke, but there's also, is it No, I feel like I'm putting.
It's a hemorrhage.
Hemorrhagic stroke makes sense.
Yeah, I don't know why
you bleed so much. Yeah.
Right. So like the vessel breaks, or you bleed into the brain. Which,
anyway,
yeah.
So interestingly, I didn't know this. People who have these cerebral ischemic events often have other complications, and particularly GI complications, GI bleeding, dysbiosis of the GI microbiome. That means you have this normal microbiome with lots and lots of different species in a balance, and dysbiosis occurs when one or more of them are either missing or not the right proportion and so forth. And constipation. And it's not just a consequence of the ischemia. It's often makes the ischemia worse because, as you know, if you've been listening to TWiN, there's a gut-brain axis. So I said gut and I pointed to my head. Brain-gut axis. There's communication between the brain and the gut, and in particular this microbiome via metabolites that are produced by the gut bacteria that get into the brain, as you will see today. And so if you have a dysregulated gut microbiome, you can actually aggravate the outcome of cerebral ischemia. But if maybe we could learn more about that and do something about it. That's the whole point of this paper, that let's figure out what the problem is in the gut and maybe we could fix it. Because, when you have an occluded vessel, sometimes they go in and do surgery and fix it, but just still this gut issue that you have to fix. So that's what we're trying to do here. And people have tried to do this for years, of course, right? They've tried to mess with the gut microbiome and so forth, but nobody's...
I don't think you necessarily draw a link between having a stroke and your GI tract being affected. Yeah, so I was really surprised when they found a link between the two.
But there is already, isn't there a link that's fairly well, a lot of people believe, between like non-vascular dementia, so like Alzheimer's disease or Parkinson's and the gut, or that might be like peristalsis, so not so much at least in Parkinson's maybe.
For Parkinson's, for sure, there's a gut, people say it's prodromal, so it sometimes precedes the diagnosis of Parkinson's. Yeah, and some people hypothesize that has something to do with your microbiome that might feature, that might kind of contribute or somehow has something to do with the aggregation of misfolded alpha-synuclein and stuff like that.
But bottom line is,
but
what's happening in your gut influences your brain.
Right. So I'm a little more surprised that stroke also has a link with microbiome.
Of course,
but
your brain also influences what you eat.
Right. That's
right.
true.
Right. That's true. Yeah. So it goes both ways.
It could be that the gut microbiome tells your brain it wants to eat something, and then the brain makes your hand move to the chocolate bar or something like that.
It's totally inexplicable why I can't stop eating Oreos.
Oh, do you put them in the freezer?
I do not, but I don't do that, but I hear they're good.
That's good out of the freezer. It's very good. Nice and crunchy.
I like to make them super mushy.
Yeah, they're crunchy, but it's too sweet, too much sugar.
People tried that? Who's the first person who did that?
I've done it. I've put it in the whole bag.
Oh,
put it in the freezer. And they're very good frozen.
It doesn't, first of all, it doesn't break your teeth.
No, because they're not very big, not very thick to begin with, right?
Oh, that's nuts. I got to try that.
Okay. As soon as you take it out, it begins to thaw. And then in the process of eating it. But
is it only
great.
Oreos? How about other cookies?
I haven't
I'm going to try
tried
a British custard cream.
Most things should be thawed, but these are good frozen.
Okay.
Anyway, back to the paper. Clinical trials have not really addressed systematically what part of the gut microbiome is connected with ischemia in humans. It's a hard experiment to do. But what they do in this paper, they do a series of experiments in mice to figure out which bacterium, and you already heard from the title, which it is, and then they look in people. And this is a beautiful example of how an animal study or studies can then give some clues about what to do in people. It doesn't always have to work that way. I read yesterday that most therapeutics, drugs are tested in animals before humans. 80% of drug that work in animals do not work in people.
80?
And I was thinking this morning, I wonder where he got that number from. I'd like to know the reference. Because it was a New Yorker article, and I'm a little skeptical these days about people making statements like that, right? But I think a lot of drugs don't work in people. And that's why you have to do clinical trials. If they were if animal models were perfect, we didn't have to do clinical trials.
Yeah.
And I think that number is much higher. It varies depending on which field. Inflated. And I think it'll be like astronomical for neuroscience.
That's also like assuming that the thing that they're trying to treat is exactly the same between the animal and the human, right? So if it's a different beast, maybe and also the treatment's not going to work.
The animals are different depending on the disease. Like here's a sad one. For heart disease, beagles are the ideal animal. Poor little beagles, right? They're so cute. Listen, I can't even kill a what's that invasive thing that's going around the Northeast? Can't remember. The lanternfly, the spotted lanternfly.
Oh, the one that we're all supposed to step on.
It's just a step on. I saw one.
You're supposed to, they tell you, they're like, hey, this public health emergency.
It's like...
I cannot step on an insect. I'm sorry. I saw one in the parking lot of the train station the other day, and I passed it, and then I went back because I wanted to take a picture because they're actually quite attractive. And it was gone. I said, good for you.
No. I get yelled at. Sorry.
That's so funny.
Anyway, I'm digressing too much. I'm sorry. You shouldn't let me do papers. So the first thing they do is to figure out what bacteria are associated with different cognitive performances in patients that have cerebral ischemia. So they put them into two groups upon admission to the hospital. The cognitively normal. So this is an ischemic patient who doesn't have any cognitive issue or cognitively impaired. And then they take feces and they sequence the genome of feces, and they can figure out from that all the different bacterial species that are in the gut. They're just sequencing 16S ribosomal RNA. All right, and I'm not going to go through the methods they use, but in the end, Lactobacillus is the only genus that correlated with cognitive decline. And they confirmed this by RT-PCR as well.
This is the absence, right?
The absence of...
It went down.
The Lactobacillus,
Yeah. The absence,
Not
reduction.
Eating enough yogurt.
Lactobacillus genus reduction, So there are 5 microorganisms in these patients, these cognitively impaired patients. They use so many abbreviations in this paper. You ever read a paper and you're like 2 paragraphs later you already forgot what the abbreviation is?
Yeah.
You remember all these things.
This is, I think, one of the first paper I have to have a list of all the abbreviation that they have so that I can keep track of it. Yeah.
So they have a mouse model where they induce ischemia by cerebral artery occlusion. Right? So they do it artificially. It makes the brain ischemic, and you get... it's a good model for this.
Yeah.
So they look at the microbiome of these mice, and there are 5 microorganisms that are reduced in CI patients that they look for in the mouse model, all right? And what they do is they give those bacteria to the mice before occluding their cerebral artery, okay? So middle cerebral artery occlusion is MCAO. Remember that because it's going to come back. They use it a lot. So it turns out that Lactobacillus acidophilus gave the most neuroprotection. They can measure the infarct size, right? Mm-hmm. That's pretty straightforward. They can measure motor function and cognition. So they have a variety of mouse tests, as you can imagine, because we've talked about some of them
even
before.
Survival rate. Like some of the mice died, and they look at how many of them died. Yeah, and it improves death. I mean, like it helps with not dying. Sorry.
Yeah, that's interesting.
So to see if it's actually Lactobacillus that's doing this, because maybe you put Lactobacillus in and something else grows better, who knows, or doesn't grow, they do fecal microbiota transplantation, which is FMT. And so they take mice treated with L. acidophilus, they take their feces and they give it to another mouse before they do this cerebral artery occlusion, and it also helps.
What's the time, remind me, the time between the fecal transplant and the stroke?
30 days.
30 days, yeah.
And they actually do they look at the feces of the transplanted mouse and see how long it takes for it to colonize. And it takes about 30 days. Yeah.
Interesting.
Right, and they confirm colonization. And then they use antibiotics to specifically deplete L. acidophilus. They found out that vancomycin abolishes and penicillin attenuates L. acidophilus protection. So it shows that the neuroprotection is a consequence of Lactobacillus acidophilus. So that's very cool. And I'm going to start taking Lactobacillus acidophilus.
It's going to become like statins. You know, it's like every cardiologist out there is taking statins, whether or not they need it. It's going to be like that.
Right. Can you just eat cheese?
I take already 2 other probiotics, and so this will be a third one. Maybe I could find one with all 3. If you go onto Amazon, you can find any probiotic. It's amazing. Okay. So in the mouse model, it's better to give the Lactobacillus before the MCAO. Remember what that is?
Middle cerebral artery occlusion.
That's right. Instead of after it. So that's, if this is going to work in people, that's hard. How do you know who's going to have a stroke, right? Or if it's just Lactobacillus, you could just take it all your life,
time,
right?
Yeah.
All the time.
Although there are people who has a more chronic case of kind of ischemia, like the Moa disease. Is that meow? I've forgotten what it's called already. Those are like kind of, you have a chronic constriction of your blood vessels and makes you very vulnerable to strokes. So those people might be constantly on this bacteria to help.
What is the mechanism of that? Did you look up to see?
I've forgotten what it's even called, but I did look it up, and they don't know what the mechanism is. But somehow the blood vessels keep kind of getting narrower and narrower.
Okay,
now look that one up.
So they have another mouse model, by the way, a bilateral carotid artery stenosis model. That's BCAS.
BCAS.
And it's the Lactobacillus also works, which makes sense. It's just ischemia, right? So now they want to figure out the mechanism. So they do a series of experiments that get more and more granular, as you will see, and which I understand less and less as we get closer to the histones, which is going to be the ultimate part. But so the LA treatment, can I use LA instead of saying acidophilus? LA treatment in these MCAO mice improves survival, it reduces weight loss, and then they do some sections, right? They look at histology. It reduces apoptosis and loss of myelin, and it restores the expression of synapse and tight junction proteins. So it's helping synapses as well. And there's also a defect in the blood-brain barrier in these patients, and this alleviates that, this lactobacillus treatment.
Yeah.
It's a wonder drug.
It's just amazing. I am actually amazed at everything they say they're going to do. It works, right? We tried this and it worked. Actually, not everything works. But. All right. So then they looked at neuroinflammation, which happens after stroke. So neuroinflammation is bad, right? Because things swell and get damaged. So they find that the lactobacillus reduces the density of pro-inflammatory microglia and increases anti-inflammatory microglia in the regions around the infarct.
Right.
Isn't that cool? Mm-hmm. So suppresses pro-inflammatory and induces anti-inflammatory. Remember that because it's going to come back as well. And of course, this also correlates with suppression of pro-inflammatory proteins, which we know a lot of, suppression of and promoting anti-inflammatory markers in the microglia. Also, cytokines are changed by lactobacillus. It reduces pro-inflammatory cytokines like IL-1 and TNF, and it elevates anti-inflammatory cytokines like IL-10 and TGF-beta-1, right?
Yeah.
Go ahead, Tim.
Sorry, they talk about they touch a little bit on how the microglia switching polarity from the pro-inflammatory to anti-inflammatory kind of phenotype. And I remember Vivian talking about this paper, saying that might be too simplistic way of describing things. But
I thought you were going to use the word tirade. You were talking about me. Yeah. But sometimes it is the easiest way to.
Yeah, exactly. Like for the purpose of this paper, I think it's an easier way to think about that. It's just anti versus pro. Yeah. Right.
Because you can only, with the methods that we currently have access to, we can't really look at microglia from all the different directions. I mean, the only way we can do that really is with, all the omics data, but that, sometimes we can't really like show that. You can't like stain or, isolate the cells and do various, kind of wet lab downstream assays that will match that. So you kind of have to choose markers that everybody knows, that we've all kind of agreed are like hallmarks of cells that are like more or less going to be inflammatory or more or less going to be anti-inflammatory. And then, maybe there'll be more details in future papers about, what specifically is important about or different about this population or that population.
Mm-hmm.
So also these mice, they looked at the behavior. They showed accelerated tape removal. Do you know what that is? They put tape on them and they take it off.
For motor function. Like, do you have fine motor skill? So if you have any pets at home, I've seen videos of people doing this on cats. You put some tape on their paw and they would look at it kind of like a philosopher in deep thought about what has happened to me. And then eventually they try to remove the tape. And you can do the same with rodents and sadistic researchers sometimes do it as a way of testing the motor function.
Not to be confused with putting paint or tape on the forehead of the mouse, which, then you're testing whether or not they have a concept of self.
Exactly.
We did that paper, didn't we, here on TWiN?
We did do that paper a while
ago. We
ago.
did. Fascinating.
So they have accelerated tape removal, reduced anxiety-like behavior. So they have increased central activity in open fields, longer open arm time, and elevated plus maze, improved novel object recognition, and reduced depressive behavior.
And that's all relative to the animal that was stroked, but not
treated with lactobacillus.
Did they do a wild type, like did they do like a sham group where they open it up and then close it again without actually doing the.
They do have sham groups.
Okay,
and those guys are like much better in all these.
Okay, so it's not like I asked this just because I think it's it's easy to think like, oh, this thing like saved the saved the stroked animals, and it's like, well, sure, relative to the no treatment stroked animals, but then it doesn't maybe bring the animal back to like the sham levels.
Yeah, I completely agree. If you look at the graph, sorry.
No, I was just going to say for a patient, it probably doesn't matter, right? Like a 1%, retention of cognition or motor skills going to count. But
I think every little probably does help. Yeah, but if you look at the graphs, I think like for the animal studies, the improvement is like maybe 10 to 20%. So it's not like a miracle cure for sure.
Yeah, right.
Yeah.
So they had previously, in these mice that they induce ischemia, they see intestinal alterations as well. So they want to know what's going on there. So they do 16S ribosomal RNA sequencing of the gut, of the fecal matter to see what's going on with the microbiome. And again, these MCAO mice have a disrupted microbiome. And it seems that treatment with Lactobacillus changes this disruption. And it makes it closer to the sham groups by already day 1 to 3 after the stroke induction. And they can tell which taxa go up, like Lactobacillus, bifidobacteria, et cetera, and pathogens like Shigella go down as well.
So walk me through the timing again here. They do the stroke. Can you walk us through that again, like the kind of order of operations of the experiment? Like
this is this is post-cerebral ischemic
treatment with LA.
Treatment, right, which is it's not as good as pre, but it still does things.
Yeah, it's just interesting to think that after the animals have the stroke, they're, some of them are very sick, like Tim said, some of them don't make it, and you have to monitor their health. So look at weight and body conditions. So like, are they dehydrated? How much weight are they losing? And then they have thresholds. Like if the animal has lost 20% of its body weight, then they have to euthanize. I don't know about this particular paper, but that might lead you to euthanize the animal and remove them from the study. But so these animals are not eating probably immediately after the stroke. So it's interesting to think a good control might have been to have a sham animal your sham animals probably, I'm not sure, not going to have the same not going to have the same sickness or motor deficits. So they're going to be eating and drinking. So their microbiome is kind of continuing to be exposed to normal, it's business as usual. So if you had a sham group, which also then had a food restriction where they were not allowed to eat or drink for 3 days or whatever the stroke mice were showing, that would probably be maybe not the most important control, but I think an interesting control nonetheless, because the animals would be taking food in, which, there's a relationship, like we were saying, between what you eat and. Yeah.
So to answer your question, the study design is they do the artery occlusion, and then at day 1, 2, and 3 afterwards, they give them the Lactobacillus, because in real world, you know that you have a stroke and you could start treating right away, right, with Lactobacillus. And they see an effect up to 3 days. They didn't look any longer than that because I don't think there's any need to, right? And they see the effect up to 3 days. It also restores intestinal function. There's a barrier disruption caused by this stroke, which you can see on histology, and occludin expression, and that's the tight junction protein. It improves gastric emptying, intestinal transit. It reduces intestinal permeability. So these are all things that happen after stroke. Oh my gosh. Reduction of gastric emptying and intestinal transit, and oh my gosh.
If you look at some of the histology of the gut, the villi of the intestine are completely destroyed, which is very surprising within like 3 days, which I think like starvation, I don't think would.
Yeah, it's like a sloughing off of the cells that create that, and then leading to this like big structural change. Is that like what do you think happens? I mean, this whole like changes, and so like gastric emptying and all of these things that are happening in the gut are autonomic nervous system regulated, and so like having a stroke is like incredibly stressful, right? So I'm actually not surprised that there would be like this autonomic dysregulation, and it's like probably not useful to be eating at that time. So it's like, okay, I can kind of see that this would, that this huge disruption is having these effects on the autonomic nervous system as well.
All right, now some mechanism. You want to know what's going on. So why does Lactobacillus work? So this is all about metabolites. This is how the gut microbiome is linked to other places, not just the brain, but everywhere else. So they did metabolomics. Okay, so you can do RNA sequencing, which everyone does, but you can also take mixtures and put them in a mass spec and figure out all the molecules that are there. So non-targeted metabolomics screening of the intestinal contents of rodents treated with lactobacillus. All right, and what they find is a big change in the composition of the metabolites in the gut of both the MCAO mice and the lactobacillus treated MCAO mice. And here we now have to get into some biochemistry. So you're going to have to listen and remember. Here, these metabolites, alpha linoleic acid.
That sounds familiar. I'm actually not joking. I don't know why.
They abbreviated ALA, which is too bad because that to me immediately meant alanine. So it's wrong. And then gamma linoleic acid, N-acetylserine, protocatechuic acid, pimelic acid, and a bunch of others are elevated after L-acidophilus treatment compared with the untreated group. On the other hand, polyunsaturated fatty acids, PUFAs, that's ALA and GLA, right, were decreased after the stroke compared with the sham group. So when you get a stroke, those go down. What I just told you was lactobacillus treatment elevates them. But these things, ALA and GLA, these linoleic acids, if you just have a stroke, they go down. And then when you treat with lactobacillus, it returns their levels to pre-stroke levels. Mm-hmm. So that's very interesting, right?
Remind me, what was the input of this experiment?
The input?
Yeah. Was it
I think it's blood.
Is it blood? Okay.
No, intestinal contents.
Intestinal contents.
Oh, okay. Oh, well, that's interesting. But I think to Tim's, whether he knew he was making it or not, what's in the gut has to make it to the brain somehow,
I think we're going to look at the blood in the next paragraph.
Yeah, that's right. No. I jumped the gun.
So this tells us that there's an enrichment in pathways that lead to ALA and Lin-A, right? And then also beta oxidation of fatty acids, whatever those pathways are. We know what they are, actually. We know all these metabolic pathways. So medium and long-chain fatty acids are affected mostly by lactobacillus treatment, okay? Medium and long-chain fatty acids. So linoleic acids, remember that. If not, we'll remind you. So as was just said, fatty acids in the intestine get to the brain through the bloodstream. So you should find it in the blood. And they looked and they found these fatty acids. After lactobacillus treatment, they see an increase in these fatty acids, both in the plasma and in brain tissues. So just as you would expect if something's going from the gut to the brain, right? And the levels of Lin-A, which is linoleic acid, and so the levels of Lin-A in the plasma and brain tissue were higher than ALA, and Lin-A tens to hundreds of times more abundant than ALA, which I'll remind you is alpha linoleic acid. Okay, hundreds of times higher. So they call this an overwhelming abundance of Lin-A, and they say this has to be important. So much of it must be the key metabolite that is mediating the protective effects of Lactobacillus and specifically neuroprotective
Question.
Effects, right? Yes.
For the brain, what did they do? Did they take whole brain? Did they microdissect regions?
They took the region around the
peri-infarct.
Yes, yeah. Peri-infarct, yes.
The penumbra.
The penumbra.
I used to work in a stroke lab briefly, so the MCAO and BCAS and penumbra.
Oh, that just rolls off your tongue.
Mm-hmm. Yep.
No. So that's
I somehow avoided having to learn how to do it. But
so that's actually the technical term.
The what?
The penumbra.
Yeah, it's like the
shadow
it's
of the
not
stroke, right?
Yeah, it's not the.
Okay.
I don't believe it's the infarct. Well, no, the penumbra is what's left behind, I think.
Okay.
I think
but
that
penumbra means shadow, of course, right?
Right.
I think I've really heard that in relation to eclipse.
Yes.
I don't know. It's also used neurologically, but here we go.
Mm-hmm.
So what is linoleic acid? It is an essential fatty acid that we cannot make. We have to eat it. It's in our diet. They eliminated, therefore, Lin-A from the diet of mice who had gotten MCAO and Lactobacillus. And if you don't feed them Lin-A, there's no protective effect of Lactobacillus against stroke. So that's beautiful, right?
Yeah.
Lin-A is clearly a key metabolite.
Lactobacillus acts via Lin-A somehow.
Yes. And it's beautiful that you can just take it away. You make a null by just taking it out of the food. That's beautiful. It's not often that you can do that, right?
Mm-hmm.
Yeah.
You don't have to make a knockout or anything. You just take it out of their food. It's amazing.
So easy.
Then what they could do is add it back, right? If you take it out of the food, you add it back, and they tried different routes. And here I got a little confused. So if you give it intravenously, the Lin-A, right? So these mice don't have it in their diet. That recapitulates the Lactobacillus treatment. But if you feed it to them, it does not work.
Yep.
So that actually makes sense.
So the point of this experiment is they ask if Lin-A is so important to the effect of Lactobacillus acidophilus, LA, the bug, the bacteria. Can we just give Lin-A and would that mimic the Lactobacillus? Right. And they found if you feed the bug, if you feed the Lin-A, the fatty acid, it didn't work. But if you inject it directly into the blood vein, it did work. Mm-hmm. So their hypothesis is that the Lactobacillus' role is to mediate the transfer of Lin-A from the gut to the blood.
Right, but why can't you feed it to them then?
So without the Lactobacillus, the transport of Lin-A from the gut doesn't work. So the absorption from the gut to the blood doesn't work very well. Right. It's what the hypothesis.
This is why I can't do any podcast on my own. You need help.
I feel like we all have, I have that, every paper that I do, I'm like, and then there was this figure where I'm not really sure what's happening.
Yeah, but I don't know whether they prove that by doing the blood works to show that Lin-A absorption get increased by the Lactobacillus. But I think that's the hypothesis.
That's right. There's no Lactobacillus there. They say this demonstrates that Lin-A could only exert a neuroprotective effect in the presence of Lactobacillus unless you give it IV, in which case that's the part that Lactobacillus does, get it into the blood, right? It says the Lactobacillus might augment the intestinal absorption of exogenously ingested Lin-A. So I guess they could put Lactobacillus in there in a Lin-A free diet and then then feed it, and that should work, right?
Mm-hmm.
Yeah.
Yes.
Yeah. But that would just be the original diet.
It's true.
But yes.
Then they gave them radioactive Lin-A in their food. So they replaced the regular unlabeled Lin-A with radioactive Lin-A. And I don't know how you're going to take care of these mice afterwards, boy, but they're radioactive. It's C13, though. I don't know. I've never heard of C13. C14 is the one that lasts forever. Many thousands of years.
Maybe C13 doesn't last forever and give you a stronger signal.
You could just keep it in the freezer for a couple of weeks. I don't know. Anyway, then they do this ischemic model, and they show that you get elevated labeled Lin-A in the plasma and the peri-infarct regions of the brain versus untreated mice that have the same diet, same food intake. Although Vivian would argue that the ones who were getting an infarct didn't eat well to begin with, right?
Yeah, but I'm going to have to go back and look at this figure.
Well,
I'm like, there's something here I want to say, but
that's a good point. That's actually a good point.
Oh yeah, where is it?
Well, despite the reduced appetite, it still worked.
It depends on the timing. If they put the radioactive Lin-A in before the infarct, before the stroke MCAO procedure. Yeah, I need to go check the notes.
Yeah. There is something else I want to say on this, but it escapes me.
Okay, you can work on that.
Maybe it'll come. Yeah, I'll think on it.
Sorry.
So let's summarize where we are so far. If you give mice Lactobacillus acidophilus, it elevates Lin-A levels in the plasma. That gets to the peri-infarct brain tissue after ischemic events. So Lin-A is the central metabolite that is explaining how Lactobacillus affects cerebral ischemia. But we're not done yet. We still have to figure out how. And now it's going to get a little harder, but we'll do it. So they do RNA sequencing on the surrounding tissue of the infarct in these mice that are treated with Lactobacillus. And they look for RNAs encoding certain proteins that go up or down, right? Mm-hmm. So they find that pathways related to inflammation and hypoxia, like TNF signaling, NF-kappa B signaling, HIF1 signaling, were downregulated in the Lactobacillus treated group. So again, this idea that Lactobacillus is inhibiting neuroinflammation.
That's interesting. The HIF1 alpha inhibition. Because
first of all,
go ahead.
Sorry,
Tom.
just quickly, they go up in the stroke MCAO mice first. So when you don't have oxygen, all these pathway that are hypoxic driven kind of go up, which is what you expect. But what is interesting is that all these pathway that go up, which you think the brain is doing in a way to maybe compensate for the lack of oxygen, they were downregulated by the Lactobacillus, which is what Vivian's going to go talk to us about.
Maybe, but because I was going to say the HIF1 alpha, when you have the ischemia, right, the brain's like, oh no, I've got to figure out some way to get blood in here. And so it uses pathways like the hypoxia-inducible factor, HIF1 alpha, to stimulate angiogenesis, which is the moya disease. It in Japanese means like puff of smoke, because when they look at images of the brains of individuals who have this disease, there's like these little, it's like a bunch of smaller vessels that sprout off of the main vessels to get blood into regions, like when the blood flow is reduced due to stenosis or narrowing of these like large arteries that feed the brain. So I looked that up. But so like HIF1 alpha is probably like kind of high all the time. And one of the things that's tricky about ischemic stroke is that you can have something, and again, the name is escaping me, but it's like reperfusion neurotoxicity or something like that. Because once blood gets back into that area, it actually is like disastrous. It creates like tons of radical oxygen species and causes major cell damage. And so it's, in a way, it's like, maybe the LA treatment is being like, okay, like let's mitigate the reperfusion injury. Because the reperfusion injury, so like you have the infarct and then you have the peri-infarct region, and you can't save anything that's in the infarct. Like it's dead. But that peri-infarct region has neurons that you could potentially save. They're stressed, but they're not necessarily going to die. And so maybe by kind of like mitigating, like pumping the brakes on these kind of like, knee-jerk reactions, so to speak, of the brain, they're able to preserve the neurons or kind of like help the neurons get through this initial, like that kind of acute, the acute phase of the stroke.
Interesting. Yeah, that might be. I didn't know about this reperfusion toxicity.
Oh yeah,
that's
because that's one of the like I said, it's tricky when somebody has stroke and they make it to the hospital. You can give them blood thinners, and you can also like go in surgically and remove it. But if you wait to do that, then the reperfusion injury is like even worse. So if you do it in a short time span before like all of these inflammatory pathways have gotten a chance to get rolling, then like the reperfusion injury is not as bad. But you don't often get it in that window.
Right.
Interesting. So it's driven by the sudden rise of oxygen when
once it's been reperfused.
Okay. Wow.
Mm-hmm.
In addition to reducing inflammation and these pathways related to inflammation and hypoxia, they see pathways related to peroxisome metabolism.
Just fat metabolism, right?
Sorry?
Oh, go ahead.
No, what were you going to say?
I was going to say it's involved in like fat rate, like processing
they are
fats, right?
Membranous organelles that are involved in beta oxidation of very long fatty acids and branched chain. That would be to get energy, right? The oxidation of fatty acids gives you energy. And so these were enriched, these peroxisomes were enriched. Pathways involved in the peroxisomes, this is being revealed by RNA-seq analysis, are enriched in the MCAO treated with lactobacillus. So they're saying maybe peroxisomes also confer neuroprotection. So we have anti-inflammation and maybe upregulation of beta oxidation in peroxisomes. Okay, so then they're going to pursue this peroxisome idea. They looked at the numbers of peroxisomes in ischemic brain tissue by transmission electron microscopy. They see an increase in the number of peroxisomes in the surrounding cells in lactobacillus-treated MCAO mice compared with the untreated group. And guess who has the biggest increase in peroxisomes in this region? Vivian.
The microglia.
Yeah, the microglia.
Which actually makes a ton of sense.
Yeah.
A ton of sense. Right, because there's a lot of cell death. And so there's all this stuff, and like, so all this debris, dead cell debris. And so the microglia are going to be, I've said it on here before many, 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 many months ago, probably, that microglia are like first responders and they're also like garbage truck men because they pick up stuff and they get rid of it. But it's actually like, it's funny. I was always thinking about it as just clearance of debris. But Vincent, when you said, beta oxidation of lipids, getting energy, I'm like, aha. These microglia, I would like to know where the material where is it possible that the microglia are becoming a source of energy somehow for cells in that peri-infarct region that might be stressed. I don't know, because there's some interest in like oligodendrocytes and astrocytes as being alternative kind of nutrients or energy sources for neurons. And so, why not microglia?
So lactobacillus increases the number of peroxisomes and peroxisome-related proteins in microglia. If you add lin-A, you get even more peroxisomes. If you deplete lin-A, you get fewer peroxisomes. So
there it is.
this enhancement of lin-A by Lactobacillus so that it gets in the blood and it can get to the brain, it causes peroxisome proliferation in microglia. Okay? But we still have more ends to put together here. So what's the role of the microglia in mediating these neuroprotective effects? They do microglial depletion in MCAO mice. So I wanted to ask you, Vivian, I guess they use clodronate liposomes. So this is a peripheral thing. They must inject it into the blood, right? How does it work? Do you know?
They say ICV, sure. Which means intracerebral
ventricular.
So they're putting it right in the brain. Okay.
Into the ventricle.
Right All
right? Isn't it? No, is it in the ventricle?
Ventricles,
or is it?
in the ventricles.
Okay. Yeah.
Okay.
So those are those like fluid-filled structures in the.
Great.
Because I was going to say, usually I don't see a lot of people use that method.
Yeah.
But yeah, I think it has its benefits because the other methods like
the colony stimulating factor.
The inhibitors of the colony stimulating factor receptor, like it impacts macrophages too. And in a lot of and that can have impacts on the readouts in the brain, even though people, anyway, that's a whole nother can of worms. But anyway, yeah. So at least by putting it directly into the brain, they're avoiding any effects on the periphery.
Yeah, because what that's the idea?
Right. Okay. Yeah.
So they deplete microglia in the brain in lactobacillus treated ischemic mice. Just depleting these microglia, there's no more protective effect of lactobacillus. So the microglia are key to this neuroprotection mediated by a gut bacterium. Who would have thought that? Then they're going to inhibit microglial peroxisome function. They're going to silence a gene that's important for peroxisome function. And they're going to use an adeno-associated virus vector to do that. Okay? And they can do it in microglia. This is so they basically inhibit the function of these peroxisomes in microglia. This abolishes the effect of lactobacillus.
Just to jump in very quickly, the AAV, the adeno-associated virus, that actually wasn't cell type selective, so they couldn't.
It wasn't.
No, it inhibited all peroxisome in all cell types in the brain, I think.
Why do they say it selectively inhibits?
Let's see.
See, adeno-associated vector to selectively inhibit.
Huh.
Is this transporter specific to microglia?
That might be what they mean.
Potentially, if the promoter is correct. But they didn't put in any flock site or anything, so they didn't have any Cree line.
Okay, I was just trying to think if there was a way to do it.
Let's assume that
that.
It is selective. It abolishes the neuroprotective effect. So it means microglia are the main ones involved in neuroprotection, and they do it through peroxisomes, which are turned up by lin-A that gets there from the gut via lactobacilli. You got that? These are the trail of crumbs that we are following.
We're zooming in and in, further and further in.
Get it further and further. That's what I kind of like about this. I wonder if they had this all mapped out in their brain before they started.
I was going to say no.
Yeah, in the discussion they said this whole thing is super surprising.
It's like a novel. You map the whole thing out ahead of time. Right?
There's somebody, I can't remember who it is, but who had a talk that was about the grand facade in papers is that
it all makes sense.
It all makes sense when it's but would you want to read a paper that was and then we tried this thing, but that wasn't right. So
no,
then
you don't want to read
thing, that. And and it's all it's interesting.
But I think that's the whole point. Maybe 80% of the point of science is you have a bunch of kind of haphazard data, and you have to string a story to try to link them together.
To make them understandable.
Make them understandable and generate some new predictions.
Often things are actually done out of sequence in the way they're presented even.
Yeah.
So it's fine. Okay, so is Lin-A really, are they acting independently? So they do an experiment where they deplete microglia with these clodronate liposomes, or they use the AAV to inhibit peroxisomal functions, and then they use rosiglitazone, which is a peroxisome agonist, so it's going to increase the activity of the peroxisomes. So that after depleting microglia or inhibiting peroxisomal function, the therapeutic effects of Lin-A, which we know can improve the stroke victims, or this other PPAR agonist, were diminished. So either one diminishes the positive effects, so that you need both. You need both Lin-A and peroxisomes, which I assumed was okay, but that really proves it. All right, and more biochemistry. I was saying, where's the clinical trial? Where is it? I'm scrolling and scrolling.
Well, they got to do their due diligence first.
3 pages more before we get to the clinical trial. So they want to know how peroxisomes are altering microglial function. So what?
I'm just saying they're zooming even further inside the peroxisome.
When you thought you couldn't get any closer.
We're going to get down to the histones. We're going to get down to the histones. So they made cell lines that are dysfunctional in peroxisomes, okay? And they find that, so they can do that by knocking down a protein or overproducing a protein in microglia cells, cultured cells. Both knockdown and overexpression led to increased expression of pro-inflammatory markers after treating with lipopolysaccharide, which is a good compound to use to stimulate inflammation. And then if they give them IL-4, these cells, anti-inflammatory markers decrease, which they say means that peroxisomes play a role in regulating this phenotype transformation of microglia, right, from pro-inflammatory to anti-inflammatory.
Yeah, the polarity switching we talked about.
Polarity switching, right, that was mentioned before. So as we already said, peroxisomes are involved in oxidation of fatty acids, both alpha and beta oxidation. Also, they make other lipids, and the clearance of reactive oxygen species, ROS. ROS. Okay.
I now work in a lab where we study ROS.
Oh.
So they overproduced enzymes that are associated with these pathways that I've just mentioned. And they found that peroxisomal ROS metabolism is key for making a microglia a pro-inflammatory phenotype, right? So ROS metabolism. Reactive oxygen species are not good for cells, right? They're produced by metabolism, just like bats make a lot of reactive oxygen species when they're flying all the time. It's not good. So you have to get rid of them. And that's why ROS metabolism is involved in. But beta oxidation is crucial for the switching to an anti-inflammatory phenotype. And there, ROS didn't play any role. So ROS, pro-inflammatory, beta oxidation, anti-inflammatory.
Yeah.
Right?
And ROS is generated by the maybe it comes from a couple different sources, but largely generated from the electron transport chain in mitochondria, which is definitely like a separate organelle. So it's
it sure is
not surprising that
and separate from peroxisomes.
Yeah. Yes.
All right. So that one more experiment to make sure all this is right. So basically, if you're going to suppress peroxisomes, are you getting an increase in ROS levels? Because the ROS was found to be key for this pro-inflammatory phenotype. So they treat these cells that they have made with LPS to induce inflammation. And then they know that giving Lactobacillus can reverse this ROS elevation after MCAO. And furthermore, inhibitors of ROS can reduce the pro-inflammatory markers, right? In these dysfunctional peroxisomal dysfunctional microglia cells when you stimulate them with LPS. So in vivo, these ROS inhibitors like NAC, they alleviate the exacerbated inflammation in the mouse brain after ischemia that's turned on by the adenovirus-associated vector. So let me just summarize what all that means. ROS elimination by peroxisomes is key for preventing microglia from having a pro-inflammatory phenotype. And so ROS elimination is key to the neuroprotective effects of L. acidophilus. Mm-hmm. Although we have to figure out how that happens now. Okay, so we're going to do that.
The LA, what was it? ALA or the Lin A?
Lin A, the fatty acid.
Peroxisomes, reduction of ROS.
Reduction of ROS, right.
Okay. And then maintenance or polarizing towards away from inflammatory.
Away from anti-inflammatory,
okay.
Right.
Oh, away to anti-inflammatory.
To anti-inflammatory, but away from pro-inflammatory. Yeah.
All right, so now they want to know how these microglial peroxisomes switch when you give that lactobacillus, right? So they do single nuclei RNA sequencing on these MCAO mice treated with lactobacillus. And they look at what's going up and what's going on. And now we see enrichment in pathways associated with histone acetyl transferase binding.
Got lots of hats.
Hats. So histone acetyl transferase is going to put acetyl groups on histones. Mm-hmm. Right?
It's going to open, right?
Which is going to open up.
Open for business.
Open for business. We're going to open up the nucleosome and transcription is going to happen better.
Just quickly.
In theory.
Just for the listener, just a quick explanation of what histones are.
Good call.
So histones are these basically proteins that help your cell wind up these, I don't know how many meters long of DNA into the tiny nucleus inside the cell. Mm-hmm. And each cell, you don't express every single one of your 30,000 gene or whatever all the time. You actually express a minority of the genes. And the way it's regulated is for genes that are not expressed, the histone kind of bind very tightly to the DNA. So it kind of is in a close kind of formation, let's say. So you can't express those genes. But the way that the cell regulate gene expression, one of the way it can do it is by putting these negatively charged acetyl groups onto the histone. And since DNA itself is also negatively charged, when you have all these negative charge around, the DNA kind of unfolds out and allows you access to transcription. Being a rat psychologist, I don't know whether that explanation is any good, so good. Correct me if I'm wrong.
It's good. Okay, so to just remind you again.
Rat psychologist.
We're adding lactobacillus to these MCAO mice, and we're looking at genes in single nuclei that are turned up or down. We see increased histone acetyl transferase in the lactobacillus group. Okay, and an increase and also an increase in N-acetyl transferase activity. That's NAC. Let me remind you that NAC
is an antioxidant.
Yes, is a ROS inhibitor. Yes, and it was in the previous experiment that it NAC alleviated inflammation in these ischemic mice through inhibiting ROS. Right. So here we're seeing an uptick in activity of N-acetyl transferase. Okay. So what this suggesting is that lactobacillus treatment induces acetylation reactions in microglia and including histone acetylation, and that's going to end up changing gene expression, which we'll see in a moment. But that histone acetylation, now we have to get into the Krebs cycle, depends on acetyl-CoA as a substrate, okay? So acetyl-CoA is that's what goes into the Krebs cycle, right? Yeah.
Yeah.
Goes into the Krebs cycle. And so basically they think that you get elevated levels of these fatty acids in the peri-infarct tissues of lactobacillus treated mice. The fatty acids are substrates for peroxisomal beta oxidation, and that gives you acetyl-CoA that can now go and get involved in histone acetylation. Okay, this is your biochemistry 101, man. Or maybe 1001. I don't know. Maybe it's a level 4 course.
400. Yeah.
400. Yep. So they. I'm not going to get into these transport details here, but they do some ELISAs to show that elevated acetyl-CoA happens in microglia after you stimulate with IL-4, and the acetyl-CoA levels go down actually in cells that have peroxisomal dysfunction, which we've mentioned before. They can do that in several ways, right? So let's see if we get here's a summary of all this. Decreased fatty acid oxidation in peroxisomes and also mitochondria, which they show. They didn't talk about that. Reduces acetyl-CoA. That can be fixed by acetate supplementation or promoting histone acetylation. This can reverse the impaired transition to the anti-inflammatory phenotype. So if you give acetyl-CoA or decreased fatty acid oxidation, you can reduce the impaired transition to anti-inflammatory. So here's a mitochondria experiment. They looked at fatty acid oxidation in mitochondria. These are primary microglial cells from MCAO mice that have been treated with lactobacillus. Lactobacillus treatment didn't increase mitochondrial fatty acid beta oxidation after ischemia. So they say this highlights peroxisomal as the key, right? What's going on in the peroxisome, not the mitochondria in terms of beta oxidation. And finally, they can supplement cells that have peroxisomal dysfunction with acetate, and that it restores the production of anti-inflammatory markers that were originally screwed up due to peroxisomal dysfunction that led to decreases in acetyl-CoA. Remember, acetyl-CoA is made by fatty acid metabolism. Okay, so peroxisomal acetyl-CoA mediates histone acetylation, and they think this drives the phenotype to an anti-inflammatory phenotype. So that's because the acetyl-CoA is eventually used by metabolized to acetate that is used by acetyltransferases to modulate gene expression activity. So they did an experiment in cells and this is the last few experiments before we get to patients. They disrupt peroxisomal function in cultured microglial cells and also in sorted
microglial cell line, or?
Yeah, that one is a line and one is sorted cells from MCAO mice.
Nice.
And they actually show reduced acetylation of a histone protein in peroxisomal dysfunction cells. And in the animals, this lactobacillus treatment reduces this reduction after MCAO. And these proteins that they're finding are histones, basically, that are the target of acetylation. They also do a labeling activity. They use C13 labeled acetyl, and they can see exactly which histone protein is being modified. And this basically confirms that Lin-A contributes to histone acetylation. So that's how we traced it all the way down from lactobacillus to the brain, to the microglia, the peroxisomes, and histone acetylation that participates in this drive of the pro to an anti-inflammatory phenotype.
That's what a ride. All the way from the gut to a tiny organelle inside your microglia in the brain, down to the DNA.
And it was, and it was like, well, they looked at. I now have to go back and look at the methods. I was going to say it because, like, I had asked earlier if they microdissected regions, and you said that early on it was just that peri-infarct region.
Yeah,
and so makes total sense to look there because that's like the place where that's the kind of battleground where ground can be lost or maintained. And it would be interesting to see, like, we know that there's a particular microglial phenotype and kind of like astrocytes are changed and whatever in the regions that are right around the infarct, but you also see changes happening on the contralateral side, so the side of the brain, like in stroke, for various other things. Like you can see there's an increase in neurogenesis from the niche that surrounds the ventricles, but it's on the contralateral side, so the side that didn't receive the stroke. So I think it would also be interesting to see to what extent the Lin-A and microglial attributes are changed or not, or, in other regions, because it's not like the effects are like the impacts of the stroke are like only happening in this area that's right around. They've actually shown that there's neuronal cell death in thalamic nuclei, and like the microglia in the thalamic nuclei are changed after stroke, like strokes that are affecting largely cortical tissue. Because there's connections between the deep layers of the cortex. They're thalamocortical and cortical-thalamic projections. So it would not, they definitely don't need to do more work here. Like they've done, it's a really cool paper, really cool story. And of course, I'm like, but what about the rest? What about
There's still one more part.
Yeah.
But before that, I forgot to tell you one more thing. So they can actually do another kind of analysis, which is called CHIP, chromatin immunoprecipitation. So basically, in these cells that have impaired peroxisomal function, they can see that because of modification of certain histones, which control anti-inflammatory genes. So that I didn't say it right. They can see modifications of histones in these anti-inflammatory genes, which cause them to have lower transcription. So that's why in the defective, impaired peroxisomal function, the anti-inflammatory genes are turned off, and they can see those histones right at those promoters responsible for that, and that's what's reversed by the Lin-A. Okay, in patients, they have a clinical trial. It's amazing the way they said, to evaluate the efficacy of lactobacillus, we did a clinical trial. It's like, oh yeah, it's the next thing. It's another figure in the paper. And actually, it's going to be published separately. This is just a part. They have 39 patients with ischemia, and 22 are going to get placebo, and 17 are going to get lactobacillus. So they treat them for 3 months after the stroke, and then they assess their cognitive functions, and they show that patients in the lactobacillus group had greater cognitive improvement compared with the placebo group. And, there's a difference in the figures. It's statistically significant. 3 months. 3 months of treatment. Of course, I think they had other things going on with them as well. They also do some brain perfusion imaging on these patients. And compared with the placebo group, the lactobacillus patients had an improvement in perfusion scores in the parietal lobe, so things are flowing better, right? They also some of these patients got bypass surgery and then 3 months of lactobacillus, and they do MRA, magnetic resonance angiography, and you can see typical bypass vessels. So new vessels are developing to go around the stroke region. They get more cerebral blood flow in this area after 3 months of treatment. So basically, the lactobacillus improves cognitive dysfunction, increases cerebral blood flow in these patients after bypass surgery.
Although some of the error bars are quite big, so it seems to be quite variable.
They are, as you might expect, right? Okay, so what's missing? The gut microbiome. What about that? So they did gut microbiome and metabolite example. So you can see a very different gut microbiome in the treated patients compared to untreated patients, increased in beneficial bacteria like butyrate producers, and immunomodulatory bacteria are increased as well. And you get, of course, an increase in Lactobacillus acidophilus. In the feces, you get elevated levels of fatty acids that we talked about, long chain fatty acids, and reduced levels of medium chain fatty acids, which aren't the ones that are helpful here compared to the placebo group, and enrichment of the ALA and LIN-A metabolic pathways in the treated group. And so
that's kind of interesting, because I thought the role of the Lactobacillus acidophilus, the bacteria, is to promote the absorption, not necessarily the content in the feces. So I actually would have thought, maybe naively, because there's more absorption, you would have less left over in the feces, but they found more.
Yeah, I'm not, but they didn't, wait, did they? I can't remember. You were talking about the absorption and transfer, that like the LA has something to do with the transfer of the LIN-A from the gut to the blood.
I think that's the hypothesis.
That's the hypothesis.
Yeah.
But that they didn't.
I don't
they
think
didn't
they
really look at.
I don't think
that.
they confirmed it, but that's to explain why the IV LIN-A worked.
But they had that radioactive LIN-A, right?
Yeah, so it showed that it made it to the brain.
Was that the one where they? So they fed it to them, right? So it did make it to the brain. So it's okay. I'm just trying to piece all this together.
Yeah, that's a lot of jigsaw puzzles to piece together.
Yeah, so they actually look at LIN-A in the fecal samples, and it does go up, but maybe it needs to go up in order to be
absorbed,
transported.
Yeah.
I don't know. But it's quite interesting. It's quite amazing that this works. And in fact, they say maybe we should give LIN-A together with lactobacillus to get even more clinical benefit. So what do we know here? Short chain fatty acids are important. Lactobacillus, acidophilus, and these activate microglia and multiply the peroxisomes to be in a anti-inflammatory state.
Yeah. If you buy the entire story kind of tied up in a bow,
in a histone.
Something as simple as just having more acetyl-CoA, acetyl group, is all you need to cause your microglia to go from an pro-inflammatory to an anti-inflammatory. And that's all you need for your stroke outcome to be a bit better. But, it's one wonders whether you do need, there's more kind of complicated things going on under the hood.
It's a good question.
I mean, plus stuff that goes, that you eat, like it goes, it's not like it's a, express train to your brain, right? There's like a bunch of different stops and like different things getting on and off and like interacting, and like maybe it can't hurt to supplement. Although there's always some small part of my brain that's like, well, how do you know that it's not hurting or that it's not having some effect? Because like what you already have, what you have here is like going to interact with stuff that you're taking in.
Sure.
Yeah. Like, how do you know? So I was thinking last night, I was like, I feel like, when they, insurance companies should, or well, not insurance companies, but when you go to the doctor, they take your blood. They should probably just start doing fecal sample collection and seeing how that changes. It'd be like, for obviously there's tons of caveats, like tons. Genetics, lifestyle, diet.
Diet.
You know,
hey, you go to a different country, your microbiome changes.
Oh, totally.
Right? But that actually, that's actually one of the questions I have is, so China is probably one of the cultures, one of the places where people don't have a lot of dairy products. So one natural experiment straight away that you can do data mine right now is to compare the stroke outcome of Moa disease or whatever stroke outcome of China versus, I don't know, what's the biggest dairy consumption country in the world? I don't know, France or Switzerland with all the cheese fondue 24/7.
They don't do that. So they're more of like a meat and potatoes kind of place.
It's India.
Because of the vegetarianism and all the paneer. Oh,
India, US, and China.
China is in the top dairy consumption?
Yeah, it's the top 3, yeah.
Oh, maybe things have changed. Oh, I'll take it back. Things have changed then. Well,
Finland, let's see. That's just total consumption, but per capita, Finland has the highest rate.
Okay.
So it's different.
But they've got a lot of good things going for them anyway.
Exactly. It might be
so this is a tough study.
Childcare.
It's a tough study because there are going to be other confounding
that's how they confound
variables.
Yeah, and even within this one study, like they didn't control for I imagine they didn't control what these people were eating.
Sure.
So it clearly has an effect though. If you just look at the.
What about if you look at countries with the lowest rate of stroke?
Oh, that's another way of looking at it. Yeah.
What is that, you think?
So Ireland, Nepal, and Switzerland have the lowest stroke rates, 36 per 100,000 people.
Nepal.
And the United Arab Emirates, Macedonia, and Jordan have the highest.
No
Macedonia
idea. And Jordan. Huh, okay. I don't see. So is it Nepal in Switzerland?
Nepal, something in Switzerland.
Nepal in Switzerland, yeah.
So the two kind of alpine, high elevation countries with low oxygen.
Interesting. Well, not all not everybody's living up at those, alpine levels.
Sure. It must be the cheese then. It's either high elevation or the cheese.
There is this, just total red herring, but there was this article that I read that was kind of like was it like the Atlantic or something like that? But it was like, there was this thing in Switzerland back in the, I don't know, 1800s or whatever, where there were high rates of something that I don't know if it's called something different now, but like cretinism, which was like major problems with like thyroid development or function.
That's it. Yep.
Right?
Yep.
And it was like on the Swiss, because Switzerland, shares a lot of borders. And it was like, I can't remember if it was like with the French border or German, but it was like on the Swiss side, you had like tons of these cases. And then like just on the other side, in France or Germany, you had like none or like very few. And it was like, there was something about the kind of erosion or like, the geology of the Alpine region that was leading I know what it was. It was like water flow, like melting water from the glaciers and things like that were removing was iodine from the diet of those people.
Fascinating.
And you need iodine in order for your thyroid to work properly. I know. I don't know why I brought that up just now, but I guess it was the whole Switzerland thing. And,
it's environmental causes of like things like this.
Right.
Oh, that's.
But just remember, this lactobacillus is not going to prevent the stroke, but if you have one, it'll help you recover better.
A little bit better, yeah.
And not 100% either. So it's.
But considering it actually potentially helps with polarity of microglia, so any other diseases that has too much of a pro-inflammatory microglia might actually, might want to start looking at this and see whether the histone acetylation changes and stuff like that. So who knows where this leads to, if true.
I was just wondering if there was a connection between wine and lactobacillus. because I'm sure I've mentioned this before, but when I was in med school in Switzerland, they had a picture. They had a picture of like a normal liver, and then just like glossy and dark, and then they had a picture of a liver from somebody who is in the Valais, a particular canton in Switzerland, where they drink a lot of white wine, and it's like not super great wine. And so I was just like looking up lactobacillus and white wine, because this Chasselas is like, I think a white wine that's made there, and it says most lactobacillus species are undesirable in winemaking. And I was like, okay, you make bad white wine, get lots of lactobacillus. Maybe
some of them are good though. Acidophilus is not one of them, but they do the malolactic fermentation, right?
Yeah.
So the malic acid gives it a crappy taste, but lactic is better. So it helps, but it's not acidophilus. So if you drink a lot of wine, you're not going to help your stroke situation.
No, in fact, you'll probably make it worse.
For several reasons, not just from the microbiome point of view.
But very provocative paper. Really interesting. Good job, folks.
Good job, Vincent.
Vincent, amazing. Amazing
that was really good.
I think I got some things wrong, but you get the picture.
Yeah.
You get the picture.
That was great.
All right. That is TWiN 63. You can find the show notes at microbe.tv/TWiN. Send your questions and comments to twin@microbe.tv. If you enjoy these programs, please support us so we can do more of good science. Microbe.tv/contribute. Tim Chung is at New York University. Thank you, Tim.
Thanks, Vincent. That was quite a journey. It was fun.
It was some journey, yeah.
Epic.
Vivian Morrison is at Tulane University. Thank you, Vivian.
Thank you, guys. This was super fun.
I'm Vincent Racaniello. You can find me at microbe.tv. You've been listening to This Week in Neuroscience. Thanks for joining us. We'll be back next month.